All right. Yeah, thank you to the organizers for having me today and giving me the opportunity to talk about this collaborative work that we've been cooking up with the Bassett, Kundaji, and Bintu labs at Stanford. So the unifying theme between all three of our labs is really this desire to understand gene regulation. And to do that, we really want to understand the key components of proteins that are responsible for regulating transcription, which are these activator and repressor domains. And so throughout the past few decades, and before I was born, people have been studying activator domains for a long time. And they've come to realize that they share a lot of similar traits. You know, these activator domains tend to be very disordered. They have patches of acidic and hydrophobic residues. And because they have all these sort of similar sequence and structural features, it's been relatively possible to generate computational models to identify and predict activators. However, repressor domains are super highly diverse as a functional class. They come from many different transcription factors and chromatin remodelers. They work through many different mechanisms. And as I'm showing on this slide, they have varying levels of structure and even disorder. So previous efforts to generate computational predictions of repressor function have sort of been limited, first of all, by what I just talked about, which is that they don't share this conserved structural features. And like many of the talks before me have kind of touched upon, if they're disordered, going through the structural intermediate to predicting function doesn't really help you very much. And then if you try to bypass the structural component before predicting function, these sort of attempts have been limited by the availability of data that actually gives you a functional training set. So a lot of the data that's been available has been assays either conducted in yeast or other model organisms that maybe aren't helpful when you're predicting human function, or they're testing like adjacent assays like protein binding or catalytic activity, which aren't a direct readout of function. And so in our work, we wanted to address both of those limitations. First, by building or generating a data set of functional readouts in human cells, and then coupling that with a deep learning model that would capture all the features that were present in our data, which included structure, but additional things as well, to predict repressor activity from amino acid sequence. And then today I'm going to talk a little bit about one of our applications, which is our attempts to use our neural net to now engineer new tools or repressor domains for nuanced gene regulation. And so luckily, the first thing we needed to generate our data was an assay that would give us the measurements we were interested in. And we have developed this in previous work led by Josh Tycho between the Baskin Bintu labs and an assay called HT Recruit. And basically, I'll go through it in a little bit in this slide. So basically, what we do is we integrate this reporter construct in a safe harbor locus in human cells. And importantly, this reporter construct has a citrine fluorescence marker, which allows us to monitor repression or silencing by our domains with flow. And even more than that, it has an IgG surface marker, which allows us now to conduct this assay in high throughput. So what we do is we design a library of 300 base pair oligos encoding 80 amino acid repressor domains fused to R to R. And this R to R allows us to recruit these repressor domains just upstream our constitutively active promoter and measure their silencing in a pooled format. And so what we do then is we allow silencing to occur for a few days, and then we conduct magnetically activated cell sorting and compute the enrichments of the domains in the on and off populations to get a measurement of repressor strength for each of our mutants. And so the second thing we would need for our work is a suitably rich library that would really capture all of the information that we are interested in. And because we wanted to make predictions at amino acid resolution, we decided to take a deep mutational scanning approach. So our library, of course, consists of our wild type domains. And then, of course, we also wanted to do a saturation all by all mutagenesis on each of our domains. But because our repressors can be highly disordered, we also included some kind of like bespoke type of mutants, which are these shuffle and reversal mutants. So we take windows of either 4 or 8 amino acids along the tile of a repressor domain, and we randomly shuffle or reverse the amino acid sequence. And we were hoping that would allow us to find things that are known to be important for disordered protein function. And then, of course, we also included some double and triple substitutions to assess some higher order interactions. And then, of course, we wanted to assess repressors as a functional class. So we had to do this deep mutational scan on many different domains. And so we actually did this on 54 different repressor domains, which we sampled from different protein families that varied in their level of structure and disorder, and that we knew would have variable strengths. And this library ended up being over 100,000 elements, and I'd just like to give a shout out to the cheapest drug that allowed me to do this. And so when we look at our data, I just want to highlight that we did the screen in biological duplicate, and you can see our data sets are extremely reproducible, which we were happy about and which wasn't a given, given the size of the library. And we conducted this in K562 cells. And so each of these data points now is this 80 amino acid sequence that has a measurement of repressor strength that is the log on and off enrichment values. And so our negative controls kind of cluster at the lower end of this, which we were happy about. And then, of course, we had our wild-type mutants on the other end, which we can see span a dynamic range of different repressor strengths. So we have these HLH wild types that are moderate in function, and the crab domains, which are known to be very strong. And I'm not going to go over this too much right now, but we can talk more at my poster. But basically, we're happy to see also that our data captured biologically relevant phenomenon like identifying secondary structures or interaction interfaces that are important for function, finding modification sites in SLIMs and disordered proteins. And also, in some cases, we were even able to see that loss of function in our assay correlated with known pathogenicity. And so the second half of our project was using our data to train a neural network to predict all the important features. And this was work that was led by Akshat, who's my co-lead and co-conspirator in all this work. And so basically what he did is he borrowed, we also used the ESM embeddings from 1B and the ESM fold embeddings to capture sequence homology and structural features. And he also combined that with biochemical property encodings from the CPT model that was released a year ago. And so after combining all of these layers in our model, he performed all of the comparisons and ablation studies that showed that this actually worked best for our training set. What we can now do is we take an input amino acid sequence, which is fed into each of these layers, and basically we get this output, which is a prediction of repressor strength, which he has changed from log off space to this P off value, which is the probability of cells that are off. And I just want to highlight that that was also an important improvement because it allowed us to move out of this log space and kind of get rid of some of the errors that we would see with differences in sequencing depth between our different mutants. And so the neural net, what we did is we basically tested how well the neural net would perform for predictions of holdout sets. So we did this for all of the sets of our data, but I'm just showing a representative example here of what we saw. And so if we held out all of the data above from this ASCL1 domain, so all the wild type values and mutant values, and trained the model on all the other pieces of information in our screen, we can see how well it can actually predict these values. And so in the zero-shot setting where it has no values from ASCL1, this purple might not be very promising, but actually when you look at the values, we see that the neural net is still able to capture a lot of important features and even rankings of what are the strongest and weakest repressors and mutants in this data set. And then importantly, when we give the model just the wild type values, no other mutant values from this tile, we can see that this sort of corrects the actual PF values that we observe and that we predict from the model, which we're happy about. And so because we saw that if we give the model all this data from things that are in the training set, it's sort of able to predict things that are close by or near to what's in the training set. And if we give it, the wild type values, it can predict things that are not just relative, but absolutely correct. And so our question was, can we now use our neural net to engineer or tune repressors towards specific strengths, giving a seed sequence that the model already knows the actual value of? And so to do this, we designed a evolutionary algorithm that was powered by our neural net for the selective pressure. And so what happens when we run this is we give the model a seed sequence, which is a sequence that exists in the training data. We ask the computer to perform random in silico mutagenesis, and then we evaluate the p-off values for all of those mutants and get rid of the ones that don't go towards what we're looking for. And so we generate a new mutant population, and the cycle repeats until we sort of evolve a sequence that our model hasn't seen before, but that predicts will have a different function similar to the target function that we're looking for. And so this is just an example of how it worked in a pilot study. We asked ourselves, could we do something like improve the strength of this moderately strong RD, which is HES3, and which is very disordered, and very, very little is known about it. The only thing we know about it is that it has this WRPW slim, which is the TLE binding motif. But otherwise, could our model understand it enough to evolve it into something else? And so we asked the model to increase the p-off value of this, so strengthen the domain. And interestingly, when we constrained the model to only allowing 8 substitutions, the success rate was 100% in this really tiny training set. But when we tested and validated 5 of the things that had evolved, all 5 of them had increased repression strength compared to wild type, which we were happy about. And even more so, we were happy that the predicted p-off values seemed to be in line with the fraction of cells that we saw by flow to be off. However, when we allowed the model to do this again, and we didn't have any constraints on the number of substitutions allowed, so we allowed the evolution to continue until the model said, here's the max improved sequence. We actually found that the performance was very poor and that we couldn't accurately predict things that could increase function, and in fact, all of these actually broke. And so what we did to test this possibility that homology and sequence divergence was a limiting factor for our model was generate another mini library, which consisted of many different evolved mutants with varying levels of sequence divergence from wild type, shown here. And so basically the main takeaways from the screen was that things with lower sequence divergence, as you might expect, tended to be more successful in getting to our target, and also tended to be better predicted by the model. And we repeated this with many other target values and different seed sequences, which I can talk about more at my poster. And so with that, I'll just conclude by sharing that this is what we did. We generated this algorithm and this huge data set that we hope can be helpful to a lot of people here. And yeah, come to my poster if you want to talk about other aspects of the project. And I'd like to just thank especially Michael, Anshul, and Lakra for supporting this work and guiding us, and especially Akshat, who's been working on a lot of the modeling and computational efforts in this project.
Cool. So maybe I missed, but in the training data, it was all fused to the TET repressor DNA binding domain. And you already validated it, so we know it works out of that context, but can you give any other sort of, how dependent might it be on that?
Yeah, the context dependency is something that we do want to explore, so that is kind of one of the limitations, is that we're doing it in this R-to-R, with this R-to-R binding domain. And that's something that our labs are also working on too, where we're fusing it to DCAS9 and trying to tether it to other places and in different contexts, yeah.
I have a question. So when you were generating your data set, were you generating it with the modeling in mind, or were these done as separate endeavors? And then after that, do you have any intuition basically of if there's a potential to minimize your training set some so you didn't have to perform as many experiments but still get the same benefit from the model?
Yeah, so we did design it with the model in mind. I didn't show much data from the double and triple substitutions because it really is hard to kind of piece out what's going on with the human eye. And so things like that were included to give the model more context, and maybe it's finding patterns that we can't see. Same thing with the shuffle and disordered mutants. It becomes very complicated to look at those and say oh, this is a pattern that's being disrupted, but maybe a model is something that can better understand those patterns. So that's why we included those higher order mutants. And then for your second question, yeah, we're still very much working on that, where we're trying to hold out different training sets and see how it performs in different cases, and even assessing like how well the model can predict things that are completely outside of our training set.
Yeah, because I guess if you were to want to do this like similar endeavor with other systems, it could be nice to know what the minimal requirement is in terms of experiments to get a model that works for
Yeah.
a general case.
Yeah, totally.
Hi, great talk. I was wondering if when you had the solutions you had where you limited the number of mutations, how similar were those solutions? And then possibly are you having, since if there are multiple solutions to the problem, is that why the model is breaking down when you allow it to keep inducing more and more evolution into it?
You mean for the evolutionary algorithm? Yeah, so in a hidden slide, I have the MSAs of the mutants that we evolved. And there are similarities in what's evolved. So for example, that WRPW slim seems to be maintained in a lot of the things that we evolved. So the model understands that to be important in repression. But yeah, there are some cases where different substitutions and different combinations do pop up. And then I think I forgot your second question, but okay.
Okay, I think we can leave it there, but thank you very much, and please join me.
Thank you.